Okay, how is studying going at the moment?
At the moment, I'm nearly done with the syllabus, and it's
Okay, very good.
still some solving in classified is remaining, but I'm nearly done.
Okay, but let's see who joined. Okay, let's start with 2023 exam. I'll start with November 2023. We will not waste time. We'll start immediately. This is, so you know what we are solving. This is November 2023, paper 21. We will focus on paper 2 for the upcoming days. Now, let's start immediately. Scientists measured the concentration of sodium ions and potassium ions in the red blood cells and in the blood plasma of a group of people, and the results are shown. So you have here a table with the red blood cells, and the blood plasma, and the mean concentration of the sodium ions and the mean concentration of the potassium ions. Use the information in table 1.1 to identify and describe. So you will identify and describe the process by which potassium ions, this one, enters the red blood cells from the blood plasma. So it will be in the blood plasma and it will enter the red blood cell. So we can see it is a concentration of 4 mole per decimeter squared, cubed, and then it will enter from the from the plasma into the red blood cell. So this is so easy. How will it enter? You will identify and describe. So the identification, this is which method? When it moves from concentration of 4 into a concentration of 100. Quickly, from low to high concentration. Are you
Active
with me?
transfer.
Of
Yes.
course, this is the identification. So you will mention active transport, and this is 1 point. And then you will discuss active transport. What will you say? Quickly, this is O level.
Uh, um, Molecules move from an area of low concentration to an area of high concentration against concentration gradient uh,
Yes.
through a partially permeable membrane.
Okay. The permeable membrane. What else? We want to mention everything about active transport. What do we use? What do we need?
Co-transporter proteins.
Yes, we need carrier proteins. These
Carrier
proteins,
proteins.
they change in shape. What is the name of the change of shape?
Induced fit.
No, no, no. Induced fit, this is the mode of action of enzymes. The change of shape is called the conformational change. We took it in enzymes. Conformational change. And the most important thing is that active transport needs energy. It needs energy from respiration or ATP. Okay? So it will move against concentration gradient, against a permeable membrane. We need carrier proteins that will change in shape, and they need the energy for the change of shape. And this energy comes from respiration. Write everything. I don't want you to be very generous in your answers in paper 2 because you don't know exactly what they want. Okay, sodium ions and oxygen molecules enter red blood cells. State one similarity and one difference between the processes used by the sodium and the oxygen. Okay, we will start with the oxygen. How does the oxygen move or cross the red blood cells? This is the lesson, cell membranes. How do the gases move?
By diffusion.
Correct. Oxygen moves by which type of diffusion? Facilitated or simple diffusion?
Simple.
Yes. Oxygen gases, any gases, the oxygen and the carbon dioxide, they are non-polar and they move by simple diffusion. In other words, they will cross the membrane from high to low. They don't need a carrier protein. Now, we are talking about the sodium ions. So we will go look at the sodium ions. Sodium ions and oxygen enter red blood cells. And how do they enter red blood cells? So the sodium ions here, they are in the blood plasma and they will enter the red blood cells. So again, these are moving also from high to low concentration. So what is the similarity?
Miss, I cannot see the screen.
You cannot see the screen. 1 minute. Seriously? Oh, it was not shared anyway. 1 minute, 1 minute. Can you see it now?
Yes.
Yes.
Okay, so they're asking about the similarity and the differences between the transport of oxygen across the cell membrane and the transport of sodium. We'll go to our table, we'll see that the sodium is moving from the plasma to the red blood cells from high to low concentration. And Mariam just told me that the oxygen also moves down a concentration gradient. So what is the similarity? Both of them move from which concentration to which concentration?
High to low.
Yes. So the similarity, they both move from high to low. So they both move by diffusion. But there is one very important difference between the diffusion of oxygen and the diffusion of sodium. Mariam told me that oxygen moves by simple diffusion. In other words, this is diffusion that requires a transport protein or it does not require a transport protein. Simple diffusion.
Does
Does
not
not.
require.
It does not require gases, if this is the cell membrane, oxygen and carbon dioxide, they move across those gases by simple diffusion. And also water, by the way, study the lesson. Water either moves by simple diffusion or through proteins or transport proteins known as aquaporins. But the gases, they cross the cell membrane by simple diffusion. They don't need proteins. Now, what about the sodium ions? They are charged. So that can they simply cross the cell membrane by simple
No,
diffusion, or they need a special carrier protein?
they need carrier proteins.
Yes, they need a special carrier or transport protein. So this is which type of diffusion? Simple or facilitated diffusion?
Facilitate.
Facilitated diffusion. So here, this is the cell membrane. You need to study the lesson to answer this question. The similarity, they will both move from high to low concentration by diffusion, but the difference is that oxygen by simple diffusion. I don't need a transport protein. Sodium, these are ions. These are polar. These are charged. They cannot cross the hydrophobic layer of the cell membrane. So they need special transport proteins. Okay, good. Chloride ions move across the membrane of the human red blood cells in a process called the chloride shift. Explain why the chloride shift is important in the transport of carbon dioxide from respiring tissues. 2 marks only. So why is the chloride shift important? Do you remember this? We took in the transport in animals, the carbon dioxide plus the water. I don't know why you don't give me immediate answers. Will give me hydrogen carbonate, carbonic acid, and this carbonic acid will dissociate into what? Hydrogen ions and hydrogen carbonate ions. What is the fate of the hydrogen ions and the hydrogen carbonate ions? How many times did we say that? Can you please speak up? What is the fate of the hydrogen ions? Give me an answer. Mariam,
Hydrogen ions bind to the hemoglobin to form hemoglobinic acid.
very good. And the hydrogen carbonate ions, this is the red blood cell. These
It
move out.
it moves out of the red blood cell into the plasma.
Very good.
Therefore, chloride ions need to enter the red blood cell to prevent a change in charges.
Very good. To prevent the change in the charge, to keep the cell neutral. That's it. So to prevent the buildup of charges inside the cell, the what's this called? The hydrogen ions will bind with the hemoglobin. The hemoglobin will act as a buffer. If the hydrogen carbonate remains inside the red blood cell and doesn't go out, the red blood cell will become negatively charged. So we will, and also if it goes out, the red blood cell will become positively charged. So it goes out because it will be carried by the plasma and it will go to the lungs to be excreted. And what will come in instead? The chloride ions will move in instead. This is called the chloride shift to prevent the buildup of charge, to keep the red blood cell neutral. Adham and Noor, you did not, what's this noise? Hello? Are you with me?
I
Yes.
can hear you.
Okay, didn't we take this in transport in mammals? Where were you? Scientist? Okay, let's look at the mic scheme if you want. Where's the mic scheme? Here, hydrogen carbonate will move out of the red blood cell to be transported in the plasma, and the chloride will move in to maintain electrical neutrality so that we balance the charges to prevent the buildup of negative charge, any one of those, okay? So it's only 2 marks. Hello, are you with me? Scientists studied the uptake of a substance F by human red blood cells. The red blood cells were immersed in a solution of this substance for 30 minutes. After this time, the scientists recorded 2 observations. The cell surface membrane of the red blood cells showed infoldings or invaginations and an increase in the number of vesicles in the cytoplasm. Identify the process by which substance F entered the red blood cell. So substance F entered the red blood cell and it caused invaginations. This is which type of entrance? What's the name of this entrance?
Endocytosis.
Correct. This is endocytosis. This is easy. Of course, this needs energy. Okay, so the question is finished. So question number 1 is all about the lesson cell membrane, by the way. Let's jump to question 2. Figure 2.1 is a transmission electron micrograph showing a section of a specialized epithelial cell found in the lining of the stomach. This cell produces extracellular proteins that are released into the bloodstream. Now, this is a tricky question and a tricky picture, but the idea is that they're telling you it is a transmission electron micrograph. Transmission electron microscope is a microscope that makes you look inside the cell. So it is showing you a section of a specialized epithelial cell. So you must be looking at one of the organelles inside the cell because this is a transmission microscope. So when you see something circular like this, and it has another circular inside it, what organelle that looks like that inside the cell? What is X?
Nucleus.
Yes. By the way, this is a nucleus, and the X is the what?
Nucleolus.
Correct. Nucleolus. So this is something that you're not very familiar with, and the question is tricky, but you will get to the answer when you notice the term transmission. Transmission is one of the microscopes that we studied, and it's called transmission because we see inside the cell. So the moment they tell you that we are using a transmission microscope, know that you are looking inside. Okay? What is the function of the nucleolus? Who will remind me? It produces what? They're asking you here. Yes. Boys, do you remember? Outline the role of the structure X and structure Y. Structure X, we said it is the nucleolus. And the function of the nucleolus, it produces ribosomes. Ribosomes are produced in the nucleolus. It produces ribosomal proteins. Now, structure Y, if this is the nucleolus, Y is pointing at what inside the nucleus? Noor, can you tell me what is found inside the nucleus? What are these?
Chromosomes or DNA.
Yes, you are correct. You are correct. Chromosomes or genes that carry DNA, chromosomes. That's it. So it's an easy question. But if you don't know, if you get the labels wrong, you did not get the question itself. Okay, so pay attention to the type of microscope. The transmission inside, scanning, we're scanning the whole picture. Light microscope usually gets you the whole cell. Okay, you need to study your microscopes. Now, the cell in figure 2.1 releases ghrelin, which is a protein that acts as a cell signaling molecule. Figure 2.2 shows the sequence of amino acids in a ghrelin molecule. The amino acid serine here in position 3 in figure 2.2 has been modified by the addition of a saturated fatty acid chain. So these are the amino acid sequences, and amino acid 3 has been modified by the addition of a fatty acid. State the level of protein structure. Adham, what is the level of this protein structure? When you have a
Primary.
sequence of amino acids, correct. This is the primary protein structure. State one similarity between the structure of a saturated fatty acid molecule and an amino acid molecule. Okay, this is biological molecules. This is the fatty acid molecule, a chain of hydrocarbons with a carboxylic acid group on one end and a CH3 group on the other end, a methyl group. These are the two functional groups. Can someone remind me what is the molecular structure of the amino acid? You should know it. Mariam, Noor. You have a central carbon bonded with what? What is the first functional group in the amino acid?
Amine group.
Yes, an H2, very good at one end. And then at the other end, what is the other functional group?
COOH.
Yes, also a carboxylic acid group, COOH. And then you have a hydrogen here and the variable R group that makes the amino acids different. So what is the common? We said it in class, I remember saying it. This is the only functional group that is found in amino acids and fatty acids. The what? Which group?
The carboxylic group.
Yes, the carboxylic acid group, the COOH. So this is the similarity. So this is the only one similarity. Another similarity, they all contain carbon, hydrogen, and oxygen, but the amino acids, they have extra what?
Peptide
Mm,
bonds.
They have the, okay, and nitrogen. So the similarity, they both have the carboxylic acid group, COOH group, and they both contain carbon and hydrogen and oxygen. But the amino acids, they have nitrogen. So here, this is biological molecules. The addition of the fatty acid chain allows the ghrelin to function as a cell signaling molecule. State how the addition of this fatty acid allows a ghrelin molecule to act as a cell signaling molecule. Adham, can you remind me what is a cell signaling molecule?
A molecule which has receptors.
It binds to receptors, you mean?
Yes, yes.
Okay, so when we added this fatty acid to the ghrelin, it became a cell signaling molecule. How
Yes.
can it act as a cell signaling molecule? How can it bind to a receptor? You
No,
just
I
said
don't
it.
know.
Why don't you know? You said it. You told me that it will bind with the receptor. Doesn't it have to have a complementary shape?
Yes.
Okay, so when we add this fatty acid chain, we will modify. It will now have not a primary structure. It will have a secondary structure, a tertiary structure, a precise three-dimensional shape, and it will have a precise complementary shape that will be complementary to the what?
To
To the receptor.
the receptor.
Okay, that is complementary to the receptor. So adding the fatty acid chain no longer will make it a primary structure. It will make it a secondary structure or a tertiary structure, or it will give it a precise shape. You have to mention that the shape will change. And this precise shape will become complementary to the receptor so that it is able to bind with the receptor. Can you think of any other answers? And the binding happens with the variable. How does the binding between things? Yes, the binding always happens with the R groups, bonding between the R groups. Now let's look at the mark scheme in case we missed out anything. Okay, so here, any three from, it will affect the 3D shape. Tertiary or quaternary structure, it will form part of the hydrophobic region, allows it to bind to the receptor. We said this and we said that. It will allow ghrelin or the part of the ghrelin to have a complementary shape. We said that. Okay, so just a detail, it will interact with the hydrophobic region of the receptor. We said it will bind, help in the interaction or embed in with the hydrophobic portion. It's mentioning the hydrophobic portion of the region because it is in the cell membrane, and the cell membrane, anything to cross the cell membrane, you have to be able to cross the phospholipid bilayer in context of bringing complementary. So we said more than 3 points. We said it will change the shape, it will have a precise three-dimensional shape, and we said it will be complementary, and we said it will bind with the receptor. So we said 3 points. Okay, Suggestion that the fatty acid allowed or incorporated or attachment to a transport molecule in the plasma to reach a target cell, reference to effect of binding, triggering the reactions inside the cell. You can even continue. That's why I tell you write everything. When the cell signaling molecule binds with the receptor, what will happen inside the cell? This is in cell signaling, also cell membrane. You first have the release of a cell signaling molecule or a ligand. It will bind with a receptor, and this will lead to a transduction process. Remember, the transduction process means a series of chemical reactions or a cascade of reactions until we finally reach a response. And only the cells that have receptor molecules are the target cells. So you will write the whole story of the cell signaling. We took it also in cell membranes. Do you remember? Boys, do you remember? Can you wake up with me, please? Are you with me?
Yes.
Okay. Now, scientists have discovered that the gene coding for ghrelin is 5,000 base pairs. This is a much larger number of base pairs that is needed to code for the ghrelin molecule shown in figure 2.2. Calculate the percentage of base pairs found in the gene that codes for the ghrelin molecule shown in figure 2.2. Show your working and give your answer to one decimal place. Okay, how will you answer this question? Noor, what will you do? This is a very tricky question. You should highlight it. What will you do?
5,000 divided 3.
3. 3 is the number of bases for one amino acid. Let's go to figure 2.2. They tell you in the question to go to figure 2.2 two times. I'll go to figure 2.2. How many amino acids do I have? They already counted it for you. How many?
28.
28. And don't forget this part, that in any protein, there is always a start codon and an ending codon. And each codon was coded for by 3 bases also. So I have a start and I have a stop codon. But they are removed when we are modifying the protein. So if we have 28 bases and we add 2, how many bases do we have? We have 28 amino acids plus 2 amino acids. We will have 30 amino acids. Every amino acid is coded for by how many bases?
3.
3.
3. So 30 multiplied by 3. What is the total number of bases?
9.
90. Okay, if I want to know the percentage, 90 of 5,000, what do we do?
90 divided 1000 times 100.
Correct. 90 over 5,000 times 100. Let me see what is the answer. 90 divided by 5, 1, 2, 3, times 100. This is
1.8.
1.8. Now let's see if we are here making up biology or we are correct. Yes, the answer is 1.8. Okay, if you don't add the start or the end codons, if you use 29 times 3 or 28 times 3, you also get 1 mark. Now this is tricky. This question is tricky. Okay, what did Noor do? He said, what did you do, Noor? You told me times 3. You only included 1 amino acid.
Yeah.
I want you to always focus and read the questions carefully, because in the question itself, they tell you shown in figure 2.2. And then again, shown in figure 2.2. So they are telling you in an indirect way, go back and look at figure 2.2. Okay, go back and look at it. When you go back and look at it, you will find that you have 28 amino acids. If 90% of you will do the 28 times 3, they will forget that we have a start codon and a stop codon. Okay, and you will still get half the grade for that. But don't forget that we always, initially, any amino acid, any polypeptide chain, we had a start codon and a stop codon, and then it is removed when it is modified. And when we change the protein and make it functional, we remove the start and we remove the stop. This is one of the modifications that is done in the Golgi body and the rough endoplasmic reticulum. Are you with me? Do you all understand how we got this question?
Yes.
30 times 3 is 90, and then we divided the 90 over 5,000. Good. The primary transcript produced from the ghrelin gene is a longer molecule than the messenger RNA found in the cytoplasm. Explain how the primary transcript is modified before translation. Now, here specifically, what are they talking about, Adham? A step that happens before translation.
Splicing.
Correct. Can you discuss what is splicing, please?
Introns are removed and exons are attached together.
Good. What are introns? The coding or the non-coding sections?
Non-coding parts. Non-coding.
Yes, continue. Non-coding sections of the
Non-coding sections.
DNA or the
is
messenger
found,
RNA. Yes. And
um,
the exons,
are
which
coding.
are the coding sections, they will be fused. That's it. And they will go out and bind with the ribosomes. Very good. Okay. Now, question 3. Plants have specialized cells for the efficient transport of assimilates. Assimilates, what are they? I'll stop here. Assimilate
Sucrose and
refers to
amino
what?
acids.
Correct. And who will carry or who will transport the glucose and the amino acids? Xylem or phloem?
Sorry.
Phloem. Very good. Table 3.1 shows some of the features of 2 different types of cell found in plant tissue, which are adapted for the efficient transport of assimilates. So the feature of the cell, it has a cytoplasm. Yes, both of them do. It has a nucleus. One doesn't, and one does. So what is cell type A? And what is cell type B? We are transporting assimilates, and one of you already said phloem, and this phloem is related to another cell which has a nucleus this time. So what is cell type A? Come on.
The
It has a few mitochondria. Huh?
sieve tube.
Yes. Cell type A is the cell in the phloem, which is the sieve tube. The sieve tube element in phloem. Correct. Identify cell type A and explain why the plasmodesmata are important. Why is the plasmodesmata important? What is the plasmodesmata anyway?
Narrow holes in cytoplasm to allow exchange of material.
Yes, they are
Without-
cytoplasmic connections between 2 plant cells. Okay? To allow the exchange of material. It allows which pathway? Symplastic or apoplastic?
Simplast.
Simplest.
Correct. Correct. So this is out of 3 marks. To mention the correct name, sieve tube in the phloem, and the explanation of the plasmodesmata. First, by the way, you can draw a diagram like this, a small diagram. This is very accessible. These are cytoplasmic connections between 2 plant cells, and they allow for the transport of material, any material, okay? Transport of glucose. For remember, when we are loading sugar and we have the companion cell, we said first the sugar will be loaded by the hydrogen pump and the co-transporter protein. Remember that? This was the apoplastic because it involves the cell wall, and then the glucose will diffuse down a concentration gradient from the cytoplasm into the phloem. So for the transport of glucose in sugar loading by diffusion. And this allows for the symplastic pathway. And you can define what is the symplastic pathway. Transport of materials through the cytoplasm and plasmodesmata. The plasmodesmata must be involved, and the plasmodesmata is only found in the phloem anyway. We don't have plasmodesmata in the xylem. Okay, so usually the answer is in the question. Read the question carefully and it will give you the answer. Any questions? Okay, now let's look at the mic scheme. Quickly. We said that. Phloem CV tube element, correct the cytoplasm. We said connect the cytoplasm, symplastic pathway, perfect. Allow diffusion of sucrose or molecules from one cell to another cell. Loading and unloading, we said all that. Any question, boys? Mariam, everything clear? B. The polysaccharide kalose is found in the cell walls of cells close to the plasmodesmata. Figure 3.1 is a simplified diagram showing you the structure of kalose. Not all hydrogen atoms are shown. So you have, this is of course very similar to the ring structure of glucose. If you study biological markers, this is glucose. This is carbon 1, 2, 3, 4, 5, and carbon 6 is outside the ring. But here, carbon 1 is bonded to carbon 3. Tell me, Mariam, what type of bond is this? Peptide or glycosidic or ester bond?
Glycosidic.
Of course, between carbon 1 and carbon 3.
3.
So what's the name of this bond?
1 to 3 glycosidic bond.
Perfect. Okay. Complete the table to compare the structure of the kalose molecule with a cellulose molecule. Okay. Feature of this polysaccharide. Monosaccharides used to synthesize the polysaccharide. In the kalose, it's beta glucose. And in the cellulose, it is also what?
Beta glucose.
Yes. Okay. Beta glucose. And write the beta like this. Don't write it like this. They consider it wrong. Now, the bonds connecting the monomers, the cellulose 1 to 4, and the kalose, Miriam just told me, what is it?
1 to 3 glycosidic bond.
Correct. Shape of the molecule, the kalose, its helix. In the cellulose, what is the molecule? Is it coiled? Is it straight? Is it branched? It is what?
Straight.
Yes, they're straight. Of course, they're straight. Orientation of the monosaccharides. In the cellulose, they are alternating. And the kalose, what do you see? They are alternating or they are not alternating?
They're not.
They're not. They're straight. They're upright. Now, this is an easy question. Hydrogen bonding is important for the movement of water through the xylem vessels in a plant. Describe the roles of the hydrogen bonding in the movement of water through the xylem vessels. This is a very easy question, direct question. Noor, can you tell me or discuss the hydrogen bonding and how does it help in the movement of water?
Hydrogen bonds allow cohesion and adhesion of water
Correct.
molecules.
Can you describe what is cohesion and what is adhesion?
Cohesion is when the cells pull each other.
Which cells? What are we talking about? We're not
قصدي
talking about
water
cells
molecules
at all.
كمان water molecules.
Yes, of course. The water molecules, when they pull each other up the xylem, creating the transpiration stream. Now, which part of the water molecule pulled on which part? There's a drawing that you can even draw.
الووتر molecule اللي فوق يعني as if it attracts الووتر molecule اللي تحتيها.
Okay, why? What is the charge on the oxygen?
Positive? لا
No.
negative.
Yes, it's electronegative. And the hydrogen, it's electropositive.
Positive.
And it will attract another oxygen molecule. The hydrogen is electropositive and the oxygen is electronegative and the hydrogen bond is here.
Mhm.
Yes,
Yes.
the hydrogen bond. You need to know the drawing. So the cohesion is the attraction between similar water molecules, between the electronegative oxygen and the electropositive hydrogen. Okay? And
Yes.
what is adhesion?
Adhesion is the pulling by the walls of the xylem.
Yes, when the hydrogen bonds are made between the water and the inner layer of the xylem, the hydrophilic layer, the lignin of the xylem. So the water can move up the xylem like this, as if it will be pulled up the xylem. Now, why are these two forces important?
um,
I
to
just told
allow
you.
water
Mm.
movement through the xylem.
Yes, so that the water column does not break, so that the water continues up the xylem and the transpiration stream. Use the term transpiration stream because this question can be 3 marks or can be 5 marks. There are a lot of things to say. You will discuss cohesion in detail. You will discuss adhesion in detail. You will discuss the advantages of these forces, that the water will move up the xylem in a continuous column. It will not break. and the water molecules will move up, and this will create the transpiration stream. And you can even add the advantages of transpiration. So it can be out of 3 marks or 6 marks. You understand?
Yes.
Okay. So you just need to write everything related. This is also the lesson plans. Water that has traveled through the xylem vessels reaches the leaves. Cooling of the leaf occurs as a result of the evaporation of water during transpiration. Water has a high latent heat of vaporization because water molecules form hydrogen bonds. With reference to hydrogen bonding, suggest why cooling of the leaf occurs as a result of evaporation of water during transpiration. Now, this is also we took in biological molecules. Mariam, can you remind me what is the latent heat of vaporization?
So I
Can
remember
someone remind me?
it is يعني high energy is needed to break down hydrogen bonds between water molecules.
Excellent. Now add something. This is 1 mark. To convert the water from what to what so that it can evaporate.
From liquid to gas, from
Yes.
water to water
This
vapor.
also biological molecules. You will define what is the latent heat of vaporization. I want to teach you how to write a neat answer. The latent heat of vaporization, you will define it. This is the heat or the energy needed to break the hydrogen bonds and convert water from liquid to gas so that it can evaporate and cool down the plant. Okay? And because the water has lots of what? Of which type of bond?
Hydrogen.
Hydrogen bond.
Hydrogen bonds. So we need lots of heat to break those hydrogen bonds and allow the water to evaporate. This heat, it will come from where? It will come from the body of the plant. It will come from the plant itself, cooling down the plant. So the plant will cool down. And this is important so that the enzymes don't nature and it doesn't overheat, etc., etc., etc. Clear?
Yes.
Yes.
Okay, good. Now, where are we? Some questions are easy, some are not. So, question 4. Now, question 4 does not look easy. Some people who are infected with HIV develop AIDS. Figure 2.1 shows the number of people that have been newly infected with HIV, new infections in 2018 across the world, and the percentage changes in the number of new infections. Now, boys, tell me, what is the difference between HIV and AIDS?
HIV is the virus, AIDS is the disease.
Yes, because you are infected with the virus and the virus remains dormant for some time. It can take up 2 years. And then when it is active, it is AIDS. Okay, now we have a graph and we have percentages and blah, blah, blah. State the full name of the pathogen HIV.
Human immunodeficiency virus.
Yes, okay. And then using only the data in figure 4.1, state what can be concluded about the change in number of new HIV infections across the world between 2010 and 2018, 4 marks. You may use the letters in figure 4.1 to identify the regions of the world. Now, I want you to focus on the question. Use only the data in figure 4.1 and the command term state. So here you will only actually just translate the data. You will not explain anything. So you will go to every country and you will mention the percentage decrease, and then you will mention the overall trend between the countries. Now look at North America. There's a 12% decrease. In Eastern Europe, 29 decrease, increase. Asia, 9% decrease. Middle East, 10% increase. So you will keep on repeating that, by the way. You have decrease, 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 decrease. Now, most of the countries, they show a decrease or an increase.
Decrease.
Most of the countries, they show a decrease, and only 2 countries show an increase, which is Eastern Europe and Central Asia, and Middle East and North Africa. That's it. This is the stating part. You will not need to do any explanation. People who develop HIV or AIDS have less T helper cells as the pathogen destroys these cells. This makes them more susceptible to TB. Explain why people with HIV or AIDS are more likely to develop TB. 4 marks. Now, this is an important question. Mariam, can you help me?
Yes, doctor.
Okay. When you have a decrease in the T helper cells, what will this do?
Uh, Less cytokines are going to be produced.
Yes, you are
Therefore,
correct. Huh.
therefore, less other types of T lymphocytes, such as the T cytotoxic and other B lymphocytes and macrophages, are stimulated.
Yes. So the immune system will be less stimulated. Very good, immune system. What else? When
This,
the immune system is not stimulated, what normally happens when it is stimulated?
when it is stimulated, the B lymphocytes differentiate to plasma cells that
Right.
produce
Stop here. Before the differentiation, we have two steps that happen before the differentiation.
colonal selection and colonal
Yes,
expansion.
this is going to be affected. So there is less clonal selection. The moment you mention that the immune system is going to be affected, then remember the normal actions of the immune system and mention that they will be affected. Normally, the immune system, when it is active, we will do clonal selection, clonal expansion, and then differentiation. All this is going to be affected. So decreased clonal selection, decreased clonal expansion. These are keywords. Less differentiation, just like you said, less development of B and T lymphocytes and T lymphocytes. B lymphocytes produce antibodies, so less production of antibodies. Less antibodies and also less what? Memory
Memory
cells.
cells.
Very good. And this example, what we are talking about is which type of immunity?
Active natural immunity.
Very good. There is a decrease in the natural active immunity. Your immune system is all of it is going to be affected. Now, when your immune system is affected, you can easily get infections like TB. And TB is an example of what type of infection that is waiting for the right chance.
Opportunistic disease.
Very good. It's an opportunistic infection. This is a straightforward question. Depends directly on how much you studied the lesson immunity. So, like Mariam said, you will decrease in the T helper activity, decrease in production of cytokines, everything else, because cytokines activates the whole immune system. It activates B lymphocytes, T lymphocytes, and macrophages. And even the activation of the macrophages will be affected. And when the B lymphocytes and the T lymphocytes are activated, they undergo clonal selection, clonal expansion, differentiation. All this will be affected. No natural immunity, and you will suffer from opportunistic infections. Okay. A person with HIV may develop a heart condition called cardiac tamponade. The pericardium is the thin sac surrounding the heart, as shown in the healthy heart in figure 4.2. So the 4.2, this is a healthy heart. When a person develops cardiac tamponade, the pericardium starts to fill with blood. This prevents the ventricles functioning efficiently. So we can see here that this pericardium, this layer is filled with blood. And this is going to affect the pressure inside the pericardium, and it's going to affect the contraction of the ventricles. Now, what do they want? People with cardiac tamponade experience a variety of symptoms, including low blood pressure and increased breathing rate. Suggest why a person with cardiac tamponade would experience symptoms of low blood pressure and increased breathing rate. Okay, not a nice question. The moment you see the word suggest, then know that this is a critical thinking question and out of syllabus. They give you information and they want you to think based on the information provided. Now, they gave you some information and they want you to know why would this patient experience symptoms of low blood pressure and increased breathing rate? Can someone think a little bit?
Low blood pressure عشان most of the blood is in the heart, not in the blood vessels.
Can you think of another answer? The pressure will increase inside the pericardium, yes, and this will apply pressure on the what?
On the atrium. صح؟
No, look at the picture. Look,
The
and
pericardium.
the pericardium, yes, the pericardium fills with blood, and this prevents the ventricles to function efficiently? The question is very clear. Will the ventricles be able to contract?
No, so مفيش blood pressure
So,
عشان ventricles won't contract.
exactly. So the ventricles, they will contract with less pressure, less force, because we are squeezing them, like they don't fill with blood anyway. The ventricles, they are under pressure. When the ventricles are under pressure, like a balloon that is under pressure, can the balloon fill with air?
No.
No. So the ventricles will not be properly filled with blood, and they will not properly pump, and they will pump with less force. They told you that. When all this happens, there will be less blood pumped throughout the body, and this will contribute to the low blood pressure. Do you understand?
Yes.
Good. When you have low blood pressure, why will this increase the breathing rate?
Because no oxygen goes to the lungs, so you need more
Very
oxygen.
Exactly. So you will increase the breathing rate to try to compensate for the lack of oxygen. Okay. So this is very logic. And this question here, when you have a question like this, read the question itself very good because the answer is in the question. Now let's look at the mark scheme in case we are just making things up. We said that there's pressure on the ventricles. The ventricles will not fill with blood. They will not pump with enough force. Blood will travel slowly. It will reduce the blood pressure. So we will have to, where are we? We answered this question, less effective, less cytokines, divide, no, yes, we said all that, good. Okay, where are we? We're here. So we will refer to compression on the ventricles. Okay, so the volume of the ventricle will reduce or the ventricles cannot fully increase in size. We said that. Less blood, we said less blood will fill the ventricles or leave the ventricles. Idea that the ventricular contraction or the systole will be less powerful. We said all this, didn't we? Yes?
Yes.
Okay. Less blood is pumped to the pulmonary or the systemic circulation. Very good. So this will decrease the blood pressure. So the faster breathing rate to compensate for the less oxygen. And we also want to get rid of the carbon dioxide that will build up. Okay. The heart or the cardiac muscle does not receive enough oxygen and glucose. So we will take the full mark for this question. Just read it carefully. Now, question 5. Finally, we're close. Figure 5.1 is a photomicrograph of a transverse section of a bronchus in the lungs. What is T? T is very easy. What is T?
Cartilage.
Yes. Okay. Cartilage. Describe the function of the smooth muscle. What is the function of any smooth muscle, Adham?
To contract.
Only
Contract.
it contracts and it stays contracted?
And relaxes.
Yes. And then this will control what?
The diameter of the bronchus.
Very good. The diameter of the bronchus, and the bronchus, what moves into the bronchus? The amount of what will be controlled?
Air.
The amount of air. Adam, your handwriting is horrible.
Yeah, I know.
No, you don't know. You have to do something about it. It is ununderstandable. The examiner will not read it. You have to do something. The
Fine.
walls of the alveoli contain some specialized epithelial cells called type 2, the pneumocytes, if you remember. These cells secrete surfactant. Surfactant helps to prevent the alveoli collapsing during breathing. Surfactant contains phospholipids, cholesterol, and protein. The components of surfactant are synthesized in the rough endoplasmic reticulum and smooth endoplasmic reticulum and then passed to the Golgi body. The surfactant that is produced is stored in secretory organelles called lamellar bodies. The surfactant in the lamellar bodies is released onto the surface of the alveolar epithelium by exocytosis. Okay, good. A lot of information. So this is the lamellar body that contains the surfactant, and here the surfactant is being released. What do they want? Each lamellar body is surrounded by a single membrane. So it is not a double membrane, a single phospholipid bilayer. Draw a diagram to show the arrangement of the phospholipid molecules in the membrane. Now, this whole exam is over cell membrane. Now, you should be able to draw the structure. You do or you don't. What is the basic molecule in the phospholipid bilayer, Noor? It's made up of what? Let's finish. Yalla, Noor.
Yes.
The phospholipid bilayer, it is made up of what mainly? What's the main molecule?
Um,
Adham.
Phospholipid head and fatty acid tails.
Yes, the
Fatty
phosphate head and the fatty acid tails, and they are facing each other.
acid.
Okay. Okay. And you can draw some structures, transport protein and another phospholipid. You can draw a peripheral protein with a with a carbohydrate chain. Please go back. You have so many pictures in the lesson, and one of them is black and white, and written next to it, you have to know how to draw it. They
Yes.
always ask you to draw the phospholipid layer, a cholesterol molecule. Okay. They did not tell you label, so just draw it. It's out of 2 marks. But this same question can be out of 4 marks. And they expect you to draw all the details. This is the carrier protein. Okay. And label. Let's see. Do they want you to label? No, just the phospholipid with head and two tails and the bilayer shown. This is only out of 2 marks. Scientists studying the production and secretion of lung surfactant have discovered that a reduction in cholesterol in the cell surface membrane of type 2 epithelial cells reduces the secretion of surfactant. Suggest why secretion of surfactant is affected by a reduction in cholesterol in the cell surface membrane of type 2 epithelial cells. Okay, another suggest question that is not nice, but it is not very hard. They're telling you there will be a decrease in the cholesterol levels, and we will not be able to carry out this process. The surfactant will not be released. Who reminds me what is the function of the cholesterol in the cell membrane actually?
Regulates fluidity of the cell surface membrane.
Excellent. It regulates fluidity. It doesn't make it more fluid or less fluid. It depends on the circumstance. So if we want to make it more fluid, it will do that. If it wants to make it less fluid, it will do that either by pulling the phospholipids together or pushing them away from each other, if you remember. Now, if the cholesterol is affected, how can the secretion of the surfactants be affected? You just gave me half the answer.
The membrane will become leaky.
You can say that, like the cholesterol will affect the fluidity. The fluidity must be mentioned. This is the keyword. And when the fluidity is affected, can the lamellar body fuse with the cell membrane and release the surfactant?
No.
No. Okay. The cell membrane has to have proper fluidity in order to carry out its functions. When the fluidity is affected, it's too fluid or too rigid, we cannot carry out the normal functions. Let's look at this. Cholesterol regulates fluidity. Membrane becomes less able to fuse. Any valid point. Yes. So what you said is correct. But mention the function of the cholesterol is that it's going to affect the fluidity. Very good. Lung surfactant is engulfed by macrophages that are in close contact with type 2. Suggest why macrophages engulf the surfactant. Now, this is so easy. Why would the macrophage engulf the surfactant? What does the surfactant contain?
Bacteria.
Yes, the surfactant, because they will stick the banana. Okay, so it might contain pathogens,
Or pathogens.
bacteria. Yes,
Bacteria.
yes, yes. Okay, it doesn't finish. The last question. I hope you noticed that paper 2 is 6 questions. Okay, state the name of the phase of the mitotic cycle during which the DNA replicates. This is easy also.
This phase.
Yes, the synthesis or the interface. During research, scientists can use modified nucleotides to prevent elongation of a polynucleotide chain during DNA replication. Figure 6.1 shows the structure of a DNA nucleotide found in the nucleus. Okay, we already know this. Figure 6.2 shows the structure of a modified nucleotide. There's a modification. What's the modification? Did you notice it? Where's the modification? What's the difference from this to this?
The oxygen was removed, the one that connects the nitrogen with the oxygen of the phosphate group.
Nitrogen? It's got nothing to do with the nitrogen. The
No,
oxygen was removed from here. Yes?
yes,
Yes. This is carbon number what?
3.
3. Okay. Now, before I move on, no, let's read the question. Suggest how the structure of the modified nucleotide prevents DNA polymerase joining it to another nucleotide. Mariam, what is the function of the DNA polymerase? Which bond does it make?
The الفوسفودايستر bond.
Correct. Now, will the phosphodiester bond be able to be made?
No.
No. Now, remind me, the phosphodiester bond, the phosphate molecule, is bonded to carbon number what and not carbon number what?
Carbon number 5 of the same group, of the same
Yes.
glucose, and the carbon number 3 in the other nucleotide.
Very good. Now, carbon number 3 has changed. Will it be able to bond with carbon number 3?
No.
No. So this is exactly talking about the structure of the phosphodiester bond. The DNA polymerase will not be able to make the phosphodiester bond, in which we have one phosphate molecule bonded to carbon number 5 and carbon number 3. Carbon number 3 has been altered. Boys, did you notice that? Carbon number 3, it was OH, it was bonding with the hydroxy group. Now it is bonding with the hydrogen. So the phosphodiester bond will not be able to make the carbon number 3 bond, and no phosphodiester bond will be made. Are you with me? Hello?
Yes.
Yes.
Okay. I will not look at the mark scheme. I want to finish. Now here. RNA aptamers are short single-stranded RNA molecules that can be used to study some infectious diseases. Scientists studying an infectious disease in animals investigated the effect of RNA aptamers on the activity of RNA polymerase that is produced by the pathogen. The aptamers bind to a specific region of the RNA polymerase. Table 6.1 shows the effect of two aptamers, F47 and F52, on the activity of the RNA polymerase produced by the pathogen. Okay, now before reading, what are these two aptamers? They bind to a specific region on the enzyme, RNA polymerase, and then they are what?
Inhibitors.
Correct. They are inhibitors. When they bind to the enzyme and they're not the substrate, then they are inhibitors, okay? Or activators, but we did not study activators. Now let's look at the aptamer present when we don't have any aptamer, and we have the F47 and the F52. And the Vmax, obviously, it decreased in all cases, and the Michaelis constant also changed. It increased. The Michaelis constant increased. Now, Noor, when the Michaelis constant increases, then this is going to affect affinity or not?
Yes.
Okay, good. The Michaelis constant will increase. It will reduce or increase affinity.
It will, when it increases, the affinity decreases.
Correct. With reference to table 6.1, compare the effect of the aptamers on the affinity of the RNA polymerase for its substrate. So, what will you do? How do you answer it? I will compare. Compare. I can make a nice line here, and I can look at the F47 inhibitor and the F52. What is the Michaelis constant in the F47? Noor, wake up with me.
508.
508. What is the Michaelis constant in F52?
523.
Good. Which will have a higher affinity?
508.
Of course. So this will have a higher affinity. Even though both of them, they have less affinity than the original, but F47 will have higher affinity. Okay? Both of them reduce the affinity, but this one will have a higher affinity. But we have to compare with the none. So I want to put the none. What is the affinity of the none? 346. So the none, when there is no inhibitor, of course, it has the highest affinity. So if we will order the affinity, this is the highest affinity, and then this has the lowest affinity. Here for everyone? With
Yes.
reference to table 6.1, suggest explanations for the effect of the presence of an aptamer on the rate of transcription catalyzed by the RNA polymerase. Mariam, can you answer this question? With reference, suggest explanations for the effect of the presence of the aptamer. What is the aptamer? And what does this do to the rate of the reaction?
Aptamer is
This
an inhibitor that decreases the rate of reaction.
Very good. It's an inhibitor. It will decrease reaction rate. Correct. We want to refer to table 6.1. Refer. Which column will you use, the Vmax or the Km?
The Vmax.
Of course, you will use the Vmax. Very easy. The Vmax, when there is none, is 1664 arbitrary units. But when we have any of the inhibitors, it decreases. And it will decrease everything else. If you remember the graph, there is a decrease in the initial rate of the reaction, a decrease in the V. Which type of inhibitor that will decrease the Vmax? Competitive or non-competitive?
A
مش
non-competitive.
الاتنين.
Yes, the non-competitive. Okay. Both of them decrease the affinity. This is a little bit confusing. Thank God they don't ask you about the type of inhibitor, because the non-competitive inhibitor does not affect the Michaelis constant, but here the Michaelis constant decreased. Okay. Now, when the reaction rate decreases, what do you say? You have a list of things that you mention. Will there be enzyme substrate complex? Will there be products produced?
No, the RNA polymerase will not bind with the template strand, therefore not forming the sugar phosphate backbone.
Yes, you are correct. It will not bind, no binding, less successful collisions, less enzyme substrate complex, less products will be produced. Okay, less reaction rate, less Vmax, and use numbers, please, because they asked you clearly to refer to table to 6.1. Mention it's an inhibitor, decreased Vmax, blah, blah, blah. Let's look at the mark scheme. This is the last question. Wow. So any three from decreased maximum rate of reaction or rate of transcription, of course, because reduced Vmax, we said it. Binding will change, of course. Binding will not happen. We did not mention the shape of the active site, but we did say that it will not be complementary. Did we mention that it will not bind, or we did not? The substrate will bind less easily. We said that. Less enzyme substrate complex. Yes, we said that. So any three from. Binding of the aptamer will prevent the substrate or the nucleotide from binding to the active site. We did not mention the active site. Binding prevents the RNA polymerase from binding to the template strand. Binding prevents the RNA polymerase to form the, you said this, Mariam, didn't you? You said it will not make the sugar phosphate backbone. Yes? So we will take the 3 out of the 3. Any valid point. We said other things. We said no collisions. We said no enzyme substrate complex. We said slower reaction. We said slower initial rate. We said slower or less products produced. So we will definitely take the 3 out of the 3. And thank God the exam is finished. This is an easy exam, by the way, easier than the one we solved yesterday. But the whole exam, 80% is cell membranes. Did you notice that?
Yes.
The lesson cell, they did not mention anything about own and immunity. That's it. This question, I thought it was tricky because most of you will not remember the start codon and the stop codon. So I highlighted this question for you. This question also is tricky because maybe you will not notice that they're talking about a transmission electron microscope and you will not be able to label those X and Y properly. The remaining questions are easy. Here it talks about active transport. Here it talks about the transport in mammals, the equation that we already know. Question number 2 is tricky, so I'll highlight it for you. And question C, this one is tricky. The remaining questions in latent heat of vaporization, if you did not study it, you'll find it tricky. You will not know, you will forget the latent heat of vaporization. This question is a suggest question. Any suggest question, there's a high chance that you do mistakes there. And the remaining questions were very easy. Also, another suggest question here. Okay. Any questions before I go?
No, thanks.
No.
Okay, of course, you will not spend the whole day doing nothing. Please solve another exam. You will not
Okay.
solve only one exam every 2 days in a week. Well, of course, I'm sure that you are solving on your own. So you should be solving at least 2 exams per day.
Okay.
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 So see you. If you have any questions, send me. Bye.